Hello, everyone. Welcome to the webinar. We'll start in approximately a couple minutes to see if more people will join us. But would like to give you all a warm welcome on this Thursday morning or afternoon or night, wherever you may be. So we'll start in a couple minutes.
Should I share my screen already then?
Yeah, you can. There are currently 41 people here. I think we can go ahead and start. So I would like to welcome everyone again to this webinar in our Noldus webinar series. I will first introduce myself. I am Huib van den Heuvel. I am a employee of Noldus. I work at the marketing department as a marketing and communication specialist for the animal products. And a bit of my background, I have a master's in animal science from the Wageningen University. And after my studies, I started working here at Noldus. But our speaker today is Dr. Irina Kovalyagina. She is a postdoctoral researcher at the Johannes Gutenberg University in Germany. And she and her colleagues recently did a study on operationalizing trait anxiety with a pipeline. And she will explain in this webinar a bit on their methodology. And before we start, I have a couple of things I would like to say. Irina, if you can go to the next slide. Just to note that this webinar is recorded. So if you are currently watching this back, you have seen that, but also if you miss something or if you want to watch it again after the webinar, we will edit it a bit and then we'll send it to all the participants here, and it will also be uploaded to our website. If you have any technical issues, I will be in the chat to answer anything. This is our first time doing a webinar with Teams, so there could be some technical hiccups, but we hope everything will go smoothly. The poll option is not available in Teams. And of course, we want to hear from you. If you have any questions, any remarks, there's a Q&A option next to the chat. and you can post any questions there. We will have 2 Q&A sessions during the webinar. Halfway through the webinar, we will answer some questions, and then at the end, we'll answer more questions. In the event that we cannot answer all the questions within the time of the webinar, we will still answer all the questions later, and we will send a document to all the participants with all the questions that have been asked and their answers in a couple of weeks. So thank you all for being here, and I will give the floor to you, Irina.
Okay, 1 second. Sorry, some technical issue, but yeah. Can you see my laser point? Yes.
Yeah, we can see your laser pointer.
Okay. Hello, everyone. Thank you for the introduction. And today I will be talking about leveraging interindividual variability in threat conditioning to model trait anxiety in unbred mice. I would like to begin with definitions. And so what is anxiety? When we colloquially speak about anxiety, and the way Cambridge Dictionary defines it as an uncomfortable feeling of nervousness or worry about something that is happening or might happen in the future. So I feel a little bit of it like right now. And in contrast, in the behavioral research, we define anxiety as a spectrum of defensive approach avoidance behaviors elicited in uncertain situations. So the main difference would be when we colloquially speak about anxiety, we emphasize the avoidance component of this behavior. And in the research lab, we mean a spectrum from approach to avoidance. So we also include the defensive approach behavior in this term. What is trait anxiety then? So we call behavior pattern a trait when it meets these 3 criteria. The criteria number 1, it is variable, this behavior variable within the group. The criteria number 2, it is consistent within the subject. And the criteria number 3, it is stable across multiple contexts. So according to this definition, trait anxiety is a stable across multiple contexts behavioral pattern biasing animal to approach or avoidance of the source of threat. And of course, we should observe some variability, high variability of the readouts within the group. Anxiety trait is a clinically relevant trait, and it is a known factor for developing stress-induced psychopathologies, meaning that when the population of humans is exposed to traumatic event, only a subset of this population will develop psychopathology. So the way humans are diagnosed with the psychopathologists is, of course, largely based on a self-report. So there are questionnaires such as state trait anxiety inventory that helps with this. And when we work with animals, unfortunately, they do not self-report, and we have to find ways to, so to say, diagnose them based on their behavior. Naturally, different tests were developed to assess mouse anxiety state. All these tests capitalize on the innate conflict of an animal between aversion to approach open, lit, and elevated spaces with innate curiosity to explore these spaces. All these models are widely used in the labs. And if we return to the definition of trait anxiety, these tests have a strong, fulfill the first criteria of this definition. So they produced variable readouts within the group. Unfortunately, repeated testing using the same apparatuses is not really possible, and the correlation between performance in different tests is relatively low. Another popular paradigm used in the labs to understand the neurobiology of defensive behavior is aversive conditioning paradigm. Conditioning is a procedure through which initial and neutral stimulus, for example, tone, acquires property of unconditioned stimulus, for example, electric foot shock, through pairing of these 2 stimuli in space and time. So whenever the animal next time presented with this stimulus, which is now a conditioned stimulus. It will elicit without any reinforcement, conditioned freezing response. Again, let's take a look at the definition of trait anxiety. So results of this paradigm, behavior results of this paradigm are remarkably consistent, and we can observe a strong freezing response in the course of 2-3 memory retrievals without significant extinction. Unfortunately, there is not so much variability of conditioned response, and therefore it's not, as it is, a very good model of anxiety behavior. As you can see, the existing paradigms only partially fulfill the definition of trait anxiety. Therefore, the goal of our project was to develop a testing pipeline allowing to operationalize trait anxiety in male and female mice. And as a majority of scientists in the field, we use inbred mice for our research. So the inbred mice coming to us from the past, and the employment of these strains is based on the belief that using animals with a homogeneous genetic background should reduce phenotypical variability. Nevertheless, it's been shown in multiple studies, including this meta-research paper, that variability of inbred animals is similar to the variability of outbred stock in both behavior and non-behavioral traits. So this is not some sort of bug, this is a feature, and we wanted to use this to our advantage to help us design a test to model trait anxiety. And to this end, we design an auditory aversive conditioning paradigm with a prolonged memory retrieval. The solution of this problem was inspired by the same idea behind why we run our Western blots and DNA gels for a longer time. So let's imagine that we have a flask of mice, and all these mice in the flask are different, but we cannot just see it by eye. And this is similar to the situation when we have a flask of DNA fragments after restriction. So what we do with the DNA fragments, we load the samples to the gel, and let's take a look at this band. So if we run our gel for 40 minutes, that appears as a single band. If we run it a bit longer, then we start getting suspicion that maybe there are more than one band. And if we run it long enough, it becomes completely obvious that there are actually two different bands in that sample. Following exactly the same logic, we extended, the time of CS presentation from 30 seconds that are standard in the field to 6 minutes. And our paradigm consists of 3 days. On the day 1, we present naive animals with a continuous 6-minute tone without any reinforcement. On the next day, these animals go through 2 training sessions, so 8 reinforcements of the shorter CSs with a mild electric food shock. And finally, 24 hours later, this animal is again exposed to the 6 minutes continuous tone. And at this point, it becomes already the CS. And now the relevant question you might have, is this paradigm fulfills the outlined criteria to measure trait anxiety in animals? So we subjected cohorts of conditioned mice to 6 minutes memory retrieval. And while we observe a homogeneous response in initial bins of the CS presentation, so each bin is 30 seconds, in both males and females, highly individual responses, freezing responses emerged in the second half of the CS presentation. So after formally quantifying the variance of freezing response, we confirmed that the variance is increasing steadily and peaks at the bin 18 and remains high thereafter. Based on this observation, we divided CS presentation into 2 phases. We call them phasic and sustained. So the phasic phase would be first 2.5 minutes of the tone presentation, and the sustained phase would be the last 3.5 minutes. The second criterion of trait anxiety was the consistency of measured behavior. To confirm that sustained freezing is consistent readout, we supplemented our pipeline with 5 additional memory retrievals performed on the consecutive days. And And in both males and females, we observe a strong correlation of initial sustained freezing response with subsequent memory retrievals. Prompted by our observation that individual animals have unique freezing responses during CS presentation, next we aim at developing a computational model to isolate animals with different endophenotypes from the total population of conditioned animals. To this end, we fitted freezing curves using exponential decay model and using parameters extracted from this model, such as intercept, decay, and together with the average sustained freezing, we clustered these animals into two groups, phasic and sustained responders. So animals belonging to both groups respond with a strong freezing to the onset of the CS, but while phasic responders return to the baseline level of freezing in the second half of the tone presentation, sustained responders continue freezing throughout all 6 minutes. And lastly, the third criterion was the stability of behavior pattern across multiple contexts. So we further extended our pipeline with a classical anxiety test battery, such as a LiDAR test, open field, novel object exploration test. And first, we observe a strong correlation of sustained freezing during memory retrieval 1 with sustained freezing during memory retrieval 2. Then sustained freezing during both retrievals correlated with the performance in an open field test. So the more mice froze during retrievals, the more time they would tend to spend on the periphery of the open field arena. And when we place a novel object in the middle of this arena, the sustained responders would approach this object less frequently than phasic responders. Remarkably, when we applied our classification on the performance of the naive animals in the elevated plus maze test, which was performed prior to the conditioning, we already could detect the differences between two groups. So the phasic responders would spend more time in the open arms of the elevated plus maze arena. So all these observations led us to the conclusion that sustained freezing is a marker of trait anxiety. I would like to emphasize that an important advantage of the fear conditioning paradigms is that they are applicable to both males and females. And while women 2 times more often than men diagnosed with anxiety disorders, translational research is predominantly using male mice and rats as a model organism. And for example, in this 10-year follow-up study of sex inclusion in the biological sciences published in eLife, it's been shown that there is no really an improvement in this situation. So the comparable number of studies in 2009 and 2019 were including females in the experimental design. And even more, so when the females are included in experimental design, only 15% of the studies actually considered sex as a variable in their analysis. And we tested this pipeline using both males and females. In line with epidemiological reports, we observed that females spend more time freezing during CS presentation in both memory retrieval 1 and memory retrieval 2 compared to males. And these differences cannot be attributed to the differences in locomotion, as females move more or comparable amount of time, move more or the same compared to males during adaptation session, so when they're naive. And additionally, we saw some endophenotype-specific differences in open field test, namely the sustained females spend more time on the periphery of the open field arena compared to sustained males. To interpret our behavior results, we used the threat imminence continuum theory. According to this theory, animals' defensive behavior is a function of perceived proximity of threat. So when the animal encounter threat in the environment, It basically has two options, depends on the availability of escape route. It will either fight the threat or flee from it. When the imminence of threat is decreasing, and then animal enters an anxiety behavior zone, which consists of post-encounter and pre-encounter behaviors. So the post-encounter behavior would be predominantly freezing behavior, and the pre-encounter behavior is a risk assessment or avoidance behavior. And finally, when the imminence of threat is lowest, the animal would perform appetitive behaviors such as grooming, for example. So how phasic and sustained responses map onto this continuum? We connected the variability of freezing responses to the variability of perceived imminence of threat. So we think that predictive value of the CS is changing throughout presentation for some animals, like for phasic responders. So from high imminence, they shift into the low imminence perception of the threat. And therefore, they map onto this pre-encounter, the sustained phase of the retrieval maps onto the pre-encounter behavior, which is risk assessment. And for sustained responders, the predictive value of the CS remains high throughout the entire presentation. And therefore, they continue freezing in this post-encounter behavioral mode throughout the entire term. Okay, and here I'm done with the first part of my talk, and I think I can take some questions.
Yeah, so if you have any questions for Irina, please post them in the Q&A. We currently don't have any questions. So we can wait a couple moments to see if any questions come in. Otherwise, we can continue. So I think we currently don't have any questions, so we can you can continue, Irina, and we'll answer maybe some questions after the second part.
Okay. Okay. So to confirm that our behavioral observations constitute real endophenotypes driven by the molecular differences between phasic and sustained responders, we performed RNA sequencing at the 2 time points, 30 minutes after memory retrieval 1 and 30 minutes after memory retrieval 2. We sequenced 4 different regions: anterior cingulate cortex, bed nucleus of stria terminalis, basolateral and central amygdala, as those regions are the key regions of the defensive circuit. We detected massive transcriptomic differences between phasic and sustained responders. More than 10% of the entire genome was differentially expressed between phasic and sustained responders, with the central amygdala having almost 3,000 genes alone. And remarkably, we detected an overlap in both time points. So in 3 regions out of 4, multiple genes were differentially expressed in after memory retrieval 1 and after memory retrieval 2. Naturally, for translational research, the most relevant question is whether the proposed model sufficiently captures features of the human disorder. So we leveraged the available data sets obtained using brain tissue from human patients diagnosed with psychiatric disorders and correlated it with our own data set. The analysis revealed significant correlations of phasic sustained transcriptome with transcriptomes of patients with schizophrenia, post-traumatic stress disorder, and bipolar disorder. Remarkably, the association of the PTSD transcriptome was stronger with the transcriptome of memory retrieval 2, which was performed at the clinically relevant 28-day time point. To further characterize transcriptomic signatures of phasic and sustained responders, we performed functional enrichment analysis of differentially expressed genes and detected multiple processes such as homeostasis, immune response, extracellular matrix organization, synapse organization, and excitation inhibition balance, and many other processes. Not surprisingly, multiple genes coding for broad variety of the ion channels, as well as the essential components of the glutamate and GABAergic systems were differentially expressed in all the regions at both time points between phasic and sustained responders. Prompted by this observation, we performed ex vivo electrophysiological recordings from the principal neurons of the lateral amygdala. While we have not identified any differences in intrinsic electric properties of these neurons, such as resting membrane potential or input resistance, the threshold of action potential generation was significantly increased in phasic compared to sustained responders. And to further understand the effect of this change on the neuronal network of the lateral amygdala, we analyzed the excitation inhibition ratio, and indeed we observed that the increase in GABA receptor-mediated currents leads to an increased inhibitory drive on sustained compared to phasic responders. And here I will summarize some key findings of our study. First, auditory aversive conditioning with prolonged retrieval sessions reveals stable inter-individual differences in freezing response. Second, individual freezing curves combined with a computational strategy allow reliable phenotyping of mice in phasic and sustained freezers across multiple batches. So for this particular study, we used over 250 animals, and in total through this pipeline, we already put, I think, more than 1,000 animals. Third, sustained freezing is a marker of trait anxiety in both male and female mice. Transcriptomic analysis of 4 brain regions at 2 time points revealed massive differences in phasic and sustained responders' transcriptomes correlating with transcriptomic signatures of post-traumatic stress disorder, schizophrenia, and bipolar disorder. And lastly, ex vivo patch clamp recordings from principal neurons of the lateral amygdala in phasic and sustained responders show significant group differences in the excitation inhibition balance. And we designed this pipeline with some reproducibility in mind. So we try to use only commercially available tools. And in case you want to adopt this pipeline in your lab, all you need is actually just a video recording system. We used ESAVision for video tracking of all our data. And yeah, basically it's only important to have a camera with a low noise and a high signal. Okay, here's some picture missing. And here I wanted to show you the setup, our setup, and I don't know where this picture went. So basically it looks like that. It's a fear conditioning chamber with a camera on top, and it's connected to the computer where there is ESAVision, so we can track online or offline. And to facilitate adoption of our pipeline, we created and published GUI based on analysis optimized in the course of this project. So these tools offer different modules for explorative analysis, clustering, dimensionality reduction, heat map representation, multivariate and pairwise correlation, and other analysis we performed in the course of this project. So this is already available online. Now we're moving to the acknowledgements. So I'd like to, of course, to thank the lab where I performed this project. It's Professor Beat Lutz lab in Mainz, and here are the labs where my collaborators are coming from, the lab of Susanne Gerber and Professor von Engelgard. And here are my co-authors, without whom this work would be not possible. And yeah, thank you for your attention. This The results of this project and what I presented today are already available as a preprint, and currently we are going through the last editorial checks at the PLOS Biology. So the whole paper should be out anytime soon. And now I will be happy to take your questions.
Yeah, so please ask away if you have any questions. Can you maybe go to the next slide real quick?
Oh,
Irina?
right.
So before we go into the last Q&A, please type your questions there. If you have any questions regarding, so for this project, Etovision was used, but if you have any questions regarding Etovision or any other Noldus products, please send me an email at the email that is shown here. Or if you have any questions about the webinar, you can also email me, or you can email Irina, whose email address I think was in the last slide. And if you want to know any more information, you can also go to our website. So yeah, and then let's go into the Q&A. I really hope that some questions come in, because we currently do not have any questions. So that means it's probably was very clear, Irina. Thank you again for doing this webinar with us. Yeah. Thank you again for doing this webinar with us. And
thank you for having me.
Yeah. Hi, Federico. And if any question comes up later, you can also email them to us, and we'll include it in the document that we will send around later. So we have a question from Stefan. I like that everyone is shifting more and more towards male and female testing. What was the hardest in doing this logistically?
Yeah, that's really great that indeed now more and more studies include males and females. Honestly, there were no difficulties for us. Working with females was even easier than with males because they can be group housed. They're not aggressive. They're just, I don't want to say much better model, but like practically they were easier to work with than males. So for us, there were no any issues working with females.
And also no logistical issues with working with the same, like male and female in the same pipeline?
So we separate the batches, and we would have a batch of females, and then we would have a batch of males. So they would not necessarily overlap with each other because our batches are quite big. It's 40 animals, the standard batch. And yeah, it's just like not possible to fit all the animals. So if you have 40 animals, 40 males and 40 females, you cannot work with the 80 animals in one day. So we'll just batch them, and yeah. So we would not create an overlap when they would be in the same rooms using the same apparatuses, females after males, but that was it. Otherwise, they were really easy to work with. And the variability that we observed in the females was a little bit less than in the males. So that made the grouping easier, and results a little bit more coherent. So in general, I can only recommend.
Okay, thank you. I think, is your question answered, Stefan? He says, but the housing condition is different from male versus female, groups versus single.
No, in this data that I presented, we used single-housed animals, but those were the first experiments in this like bigger project. And in the further experiments, we tried to group house animals, and then we group house all females and males. It's a little bit more tricky with males because when you do perform some behavioral experiments with them and then you put them back into the cage, they can start fighting, and that, I guess, some additional stress on them. Although our animals were not fighting so much either, but Yeah, in general, they were treated equally. It's just there are less problems with females. I hope that answers the question.
Does that answer your question, Stefan? Yes, thanks. Indeed answered. I have experience in group housing males and females. Feel free to ask me for tips.
Okay. Yeah, in general, we are aiming to shift to the group housing because also the regulation on this matter becomes more strict, and yeah, it's just like additional stress for the animals to be single housed.
Mhm. And if you want to ask a question in person to Irene, I think that's also possible. But then you have to raise your hand, and I can give you permission to speak because we have currently disabled all the microphones.
Our peer review was also not so many questions. Maybe.
I think, Lisa, do you have a question? We'll see if I can unmute you. You can now speak, Lisa. But you have to unmute yourself first.
Thank you. Couldn't find the Q&A. Oh, there goes.
Okay. But feel free to ask your question.
Do you... It echoes quite badly, so I'll speak slowly. It went quite fast, this comparison between the different behavioral tasks that reveal sustained anxiety or avoidance. And I was wondering if, let's say, one didn't use the specific fear conditioning task, but some other task repetitively, if you think that would also indicate the different traits of the animals.
Yeah, we actually tried that. We did some of the tests repeatedly, like open field, something else. And what we observe is that activity of the animal in a second test drops significantly, so they're not engaging with the environment anymore. So we dropped that idea, and then we created this redundant pipeline with multiple anxiety tests that are targeting all approach avoidance behavior. And the correlations that we observed in this test were between the tests would be around 0.3. So they were significantly less powerful compared to the fear conditioning, where we would see the correlations 0.6, 0.7. So that just had more power, this test, compared to the doing just anxiety tests. And then we use this anxiety test to further elaborate on the phenotype of the animals. So when we selected animals for sequencing, we would, of course, base first selection on results of the conditioning, and then we would look into other tests like open field test or EPM test and see how they perform there, and would select for this particular project, we selected animals with a consistent phenotype. So we have in the paper in all details how they were selected. And so we try to select the animals that consistently perform as avoiders or approachers. So I don't know. I feel like conditioning tasks are so much more stable and consistent compared to the anxiety tests, and they're not so difficult to perform. So I don't know if that answers your question, or was it something else? I know you're muted. Are you still muted?
I am muted. Sorry. Thank you very much for your question, Lisa. Okay. Are there any other people that have a question that they want to ask in person or? See in the chat? No. Also nothing in the chat. Well, then I think we're all out of questions. So Irina, I would like to thank you again. I would like to thank everyone for coming as well, for registering and listening to Irina. And yeah, I think we'll send you the recording in a couple of weeks probably. And yeah, we will end it there. Thank you very much.
Thank you for having me. Stay tuned for our paper.
Yeah, stay tuned for the
Okay.
paper.
Okay. Ciao.
Okay, bye-bye.
Bye.